We have finished the basic topic of biotechnology principles and tools and techniques, principles and processes, or including tools and techniques. Now we go to the second unit, which is termed as biotechnology and its applications. In biotechnology and its applications, we will be looking in terms of what are the applications of biotechnology in terms of industrial scale production. When you say industrial scale production, it could mean we are dealing with antibiotics, vaccines, enzymes, industrial products. It could also include the study of the applications of biotechnology to biopharmaceuticals. Now, biopharmaceuticals are drugs. We will be looking in terms of what are the most important drugs which are helpful for primarily for medicine. And again in BT and its applications, we would also be studying biologicals. Biologicals include enzymes, vaccines, vitamins, antibiotics, antigens, antibodies, and other new novel compounds which are useful for human welfare in terms of agriculture, in terms of medicine, and in terms of industry. So, parallelly in BT and its applications, you would also be studying about genetically modified organisms, which include microbes, plants, animals, and fungi. The reason why genetically modified organisms involve maximum microbes and fungi is because microorganisms and fungi can sporulate very fast. And the reason why we have most of the genetically modified organisms include the crops or the plants is because plants are totipotent. Totipotency is a very important property of the plant, whereby a cell can remember when to divide and a single cell can give rise to the entire sort of a plant. So, totipotency is a genetically inherent character of plants, and microbes have the capability of growing on media, and it is very easy to multiply the sort of microbes even on a given nutrient medium under sterile conditions. Thereby, whenever we are dealing with relation to applications of biotechnology, we are looking in terms of recombinant DNA. We are looking in terms of recombinant DNA, how it is taken up by means of restriction enzymes, cloning vectors onto a competent vector, and how the recombinant DNA is expressed so that it has got the sort of foreign gene of interest, and at the same time how do we culture the cells by means of plant tissue culture, which is regulated growth of the particular cell on a defined medium under certain conditions. So, the applications of biotechnology spread into various areas such as therapeuticals. Now, therapeuticals are compounds or substances, biological substances which are used in treatments. They are used in medical treatments such as drugs and vaccines. Biotechnology has got immense applications even in diagnostics. That is, if you want to exactly detect or you want to analyze or you want to trace a particular DNA or an RNA or a protein molecule, the maximum applications of biotechnology is in the area of food, because we understand we live in a sort of a world of very high population. Given high population, given changing climatic conditions, it is very important that we have got to feed the world. We cannot feed the world when there is less of soil, less of water, less of nutrients, and when crops cannot grow beyond a particular sort of an extent. Thereby, if you take genetic modification or genetically modified organisms, GMOs have got lots of application in terms of GMO crops. Like if you take, they have wider applications in agriculture. We have genetically modified rice, maize, potato, sugar, and again we have got tomatoes, genetically modified tomatoes, brinjal, soya bean, and the list goes endlessly. So, we have primarily the application of genetic biotechnology is in the field of food, that is with relation to crops, and it has got also applications in terms of processed foods. All processed foods, most of the processed foods are derived primarily from genetically modified crops. If you want to make the gluten thin, or if you want to have flavor saver tomato, that is a tomato which has got a lot of flavor and which is frost resistant and which is useful for making tomato ketchups, sauces, etc., we need to exactly get processed food from crops. So, getting of processed food from crops, and again the basic food which are enriched with vitamins, starch, etc., all these are the two important applications of biotechnology. Biotechnology again has got applications in a broader area which is termed as bioremediation. Now, bioremediation means how do you clean? It relates to cleaning up of the environment. Bioremediation is a very advanced science, and bioremediation deals with relation to treatment or cleaning up of contaminated soils, contaminated water, contaminated nuclear waste, and also with relation to all types of polluted soils and waters and also atmosphere. So, in bioremediation, we use genetically modified organisms to clean up oil spills, toxic waste, and also nuclear facilities. Biotechnology again has got lot of applications even in terms of waste treatment. For cleaning up waste as well as both nuclear waste as well as chemical waste, for cleaning up waste as well as recycling of organic compounds with the release of oxygen and other sort of an very important sort of a components in the atmosphere, waste treatment also has got a very important role. So, bioremediation, waste treatment, and biotechnology again has got applications in energy production. For production of hydrogen, which is going to be the future fuel. The future fuel on the base of which we have the entire automobile industry running in, and all clean, like if you believe in clean technology, as well as clean and green technology, biotechnology seems to be the sort of an answer. So, energy production is also a very, very important sort of field in the case of biotechnology. Next, we will primarily be viewing about which are all the critical areas in research in biotechnology. We are now focusing into what are the critical areas of research in biotechnology. It is very important when we have biotechnology as an interdisciplinary science. We understand it is emerging, or it has been emerging on one side of biology, and on the other side you have primarily learned about what are the tools and techniques of biotechnology. And we understood that we have several sort of vectors, restriction enzymes, hosts, and several tools and techniques which can enhance biotechnology. Just previously, I had just given you what is the scope of biotechnology, how biotechnology seems to influence everything with relation to the quality of life. So, if we are looking in terms of improving the sort of a quality of life, if ever we want to improve the quality of life, we need to look at two important issues. One is with relation to food production, and the other relates to health and medicine, and the third one relates to environment. So, these three are the major sort of applications of biotechnology lies in health, medicine, environment and food production. Now, if you ever want to improve the quality of life, what is the most important way how you could be doing about this? So, the first important thing which we are going to address in research is how do you identify and provide. So, you need to identify a microbe or a gene of interest. You identify a gene of interest and you provide it the sort of a gene or the microbe. You are using the gene or the microbe as the best biological catalyst. You are using it as a biological catalyst. That is, you are trying to speed up a chemical reaction. You are using it as a best biological catalyst, the one which is the best biological catalyst, or you are trying to use a sort of an enzyme, or it could be a sort of an enzyme. So, you are using a gene or a microbe or an enzyme. You are looking at it in terms of a biocatalyst and you are seeing to that you develop the world's best biocatalyst. That gene, microbe or an enzyme is the best biotechnologist which is available for the application. Thereby, your judgment is how do you identify the gene of interest, and you are looking, because we take biotechnology and look at it as a sort of a chemical process. And second, we have to know what are the optimum conditions. What are the optimum conditions developed by genetic engineering, by biotechnology, for the catalyst to act, in order that the catalyst need to speed up a chemical reaction very, very fast. What are the optimum conditions which you should provide to the sort of a catalyst? So, identifying the biological catalyst is one aspect, then providing the optimum conditions for the particular catalyst is the second option. Thirdly, we have to look at what are the current that is existing and the futuristic or improved downstream processes. The downstream processes in biotechnology. We are looking at downstream processes. We are saying what are the presently what is available, what could be available in the future, and how do we improve these sort of downstream processes. Primarily, how are we improving the downstream processes or the sort of downstream technologies? We are again looking at the big picture of the technologies. How can we improve the fermentation of insulin, the fermentation of an enzyme? How can we improve the quality of a particular food which is enriched with vitamin A in a bioreactor? So, we are looking at how do we improve the technologies so that it can lead to the purification. For the purification of the protein or the enzyme, so that we get the protein or the enzyme which is very, very pure, or the biologically derived organic molecule. So, at the end we require a sort of a molecule or a compound which is biologically derived. It should be present in the pure state. It should come out from a sort of a very appropriate as well as a very efficient downstream technology, wherein you are trying to see to that the technology is very, very efficient. It could relate to the futuristic situation or to the current situation. So, we are also looking, so there are three important research areas. One relates to the gene of interest or the microbe of interest. Next relates to what are the optimum conditions under which we need to grow the gene of interest of the microbe, and what exactly happens on the sort of a downstream processing. So, these are the three critical areas of research which always biotechnology addresses to. So, these are the three important areas because of which the quality of life can always be enhanced in terms of food production, in terms of health and environment, and health and medicine as well as environment. So, the critical areas of research primarily deals with these particular things. So, if ever we are thinking in terms of, we are asking ourselves what use is biotechnology. Biotechnology has got tremendous applications, but on each of the particular applications we have to test ourselves in terms of research, asking questions, in terms of advancements, in terms of relevance, and in terms of newer opportunities which we can create in terms of the gene, the microbe, or the sort of a process technology. Now, we would be looking in terms of various issues, like first thing is we will be knowing about BT and agriculture. In BT and agriculture, we would be looking in terms of what are the different methods of food production. Then among the food production, why is it that we are going to go in for genetically engineered crop based agriculture, and what do you exactly understand by the green revolution, and how is the green revolution different from another sort of a revolution which is recently on the upsurge. It is termed as a gene revolution. How is the gene revolution very important when compared to organic agriculture or agro based agriculture, and we will exactly know that what are the basic themes or the principles of the green revolution, and what happened or what is the attitude of the farmers in developed countries with relation to green revolution, and what exactly is the meaning of the term gene technology or genetic engineering, and how do you account for the basis of genetic engineering, especially in agriculture. So, we would be looking at this in terms of genetic modification, in terms of green revolution, BT and agriculture.